It's very nice.
Do
Okay,
you
so
mind if we record the
no, it's okay. It's
presentation?
okay. Yeah, it's
Because we
perfect.
can spread
Perfect.
we can share with other wineries after the presentation.
Exactly. Exactly. So, hello to everyone. Perhaps first, how long time do you have for this meeting? 30 minutes, something like that?
For your presentation, I calculate about 20 minutes of
20
presentation.
minutes. Okay.
Then we have some time for questions.
Okay, good. Perhaps first few words about the company. So Everyone Count is a company based in France, near Bordeaux. I think you know all this city. We are specialized in biodiversity analysis, and we intend to support the ecological transition of agriculture, but also of all the environmental actors. And our innovation is to combine advanced technologies such as artificial intelligence, like everybody else, but also eDNA, sensors, aggregation of open data. Everything is in the name. Everyone counts. The idea here is to aggregate the maximum of data in order to be able to assess the impact of land use practices on ecosystems. The ecosystem we prefer is the countryside, rural ecosystem. And we are focusing now for, I think, 2 years, especially in the wine industry and viticulture. And the other very important thing is the fact that we take the approach. We focus on two approaches. The first is the fact that we have a scientific approach. All the things we are developing, we are based on scientific publication, and the other thing is we are very data driven. And all this, both approaches are very inspired by the clinical diagnostic because I'm a former guy from the clinical diagnostics. And what is the product? So the product behind the company is a platform which is called BioAtlas, a digital platform for aggregating, monitoring, tracking, and analysis biodiversity data over time. Today for wine estates, but we also have a project with local authorities, with companies from sector like agriculture or cosmetics. And the main idea is to be able to promote nature-based solutions and regenerative practices. Okay. And to make short a long story, perhaps, I'm going to show you what the platforms looks like. And I'm going to take the example of the project, the most advanced project we have, which is called Château La Perruche. It's a vineyard near Bordeaux, where we are collecting data now for more than 80 months. Okay. So I'm going to share. Do you have questions for the introduction, or I'm sharing the screen and start with the platform? It's okay.
No, it's okay, I think.
Okay, so if I want to share, because I'm not so familiar with Zoom, I think I can do that. Do you see the map?
Yes.
Yes, okay. So this is the first. So the platform at the beginning of the platform, you have, of course, an interface to connect for every user has got his own access to the platform. And so yeah, I'm going to La Perruche. And the first thing you have is a map in order to visualize all the data we are collecting. Okay. We also have another visualization is just to see the data. Okay, I'm gonna see the data just, and we also have dashboard. Okay, to focus on special data. But I'm starting with the maps. It's a good way to explain you how we work. So here you get a map just to see the localization. So if you are familiar with France, we are here near Bordeaux. Is the southeast of Bordeaux. This is not the most famous area for Bordeaux. It's the Cadillac. I don't know if you know the appellation Cadillac, but here the owner make a great wine. I am a big fan of the product. The first data we collect is all the free data. We have a lot of free data in France, but I think the same in Chile. We have identified more than 200 databases where we can find data. It's different layer. To give you some example. So we don't furnish the 200 database. The idea is to discuss your question and then to choose the open database you need for answering to the question. So here, for example, because it's in Vignon, I'm sorry, it's in French, but it's not a problem for us to translate the interface. So to give you an idea of the kind of data, first you can have the border of the what we call the cadastre in France, the border of the vineyard. We can also collect free satellites images. In Europe, we can have more interesting satellite imaging. For instance, here it's an image where you can have the nature of the soil. So here we can see it's forest, vineyard. It's not interesting for the owner, but here we need to know what we have around, because after that we are going to make statisticals with what we get around in order to interpret the influence of what we get around on what happened in the vineyard. Okay. And because this is wine business, we also collect information about the geology, because here, as you can see, we have information about the terroir. We have 3 different terroirs, so this is open database with a layer where we have information of the nature of the geological nature of the soil. And we also have biodiversity data on this open database. The most famous one, perhaps you know, is a database which is called JBIF. Perhaps you heard about it. It's a worldwide database. You can see all the points there. On this database, you find all the data collected by the scientist, the naturalist, the school. When I don't know if you use a phone application to recognize plants or so on, so on. All this data are stocked on or storage on the cloud, and we can get them through a database like JBIF. And to give you an idea what is it, you can see the point there. So I take this one. I can go directly to the open database, or in this case, you can see it's Pestacite odorante in French, Tussilago pyrinacai, scientific name. So by this collection of open data, you have a free atlas of the biodiversity, because as you can see there, we have collected, like if you read here at the right, upright, more than 9,000 of data. And if you look into this data, half of this data concerning the flora and half of this data concerning the fauna. So with that, you have a free atlas of the biodiversity around your vineyard. So this is the open data. Because we want to correlate the impact of practices to the biodiversity, of course, we collect the practices. And it's very simple for us, because it's through interview of the owner, and then we transform the information he gave you through the interview in information that we can put on a map. So I can show you here the practices. So you see, this is point and surfaces. Yeah, if I go on the points for perhaps I can translate easy. It's hives. You also have a bird nest. You have you can see a geometrical form. It's edges of fruit trees and so on, so on. And you can see what we have collected around 40 different practices, and we are going to correct, I'm going to show you that, these practices to the biodiversity data. So we have the open source, the practices of the owner, and the last one, which is always the thing people reminding about us because this is the technology part. We also use sensors. So I show you another map here. It's the map of the sensors what we have put at La Perruche. So here at La Perruche, we have put, if I remember, something like 52 sensors. It's huge. It's too much. You don't need, it's too many sensors. You don't need that to follow 50 hectares of viticulture. It's because it's our experimental labs and we are testing many things. With 6 to 10 hotspots with video and sound recorder is sufficient to have information. So here we have put all these sensors and we use very simple non-invasive sensors. We use cameras, sound recorder, ultrasound recorder if you are interested with bats, because sometimes bats are interesting because they are considered like auxiliary for viticulture. And we also make soil sampling and analyzing by different technologies, depending on the question, okay? So we collect open data, practices, and sensors. And the third thing you have is you can just have on our platform the possibility to see the data. So I'm going to show you the video because this is the what the people and me prefer, for instance. So I don't know if I choose this one, for instance. So with the cameras, we can have this kind of videos. We have a lot of, I think we have something like 500 videos. So, the interest of the sensor is by the fact that they are working 20 years a day, on Sundays, on every time. So we are collecting the data. And on the platform, we also have another way to see the data is through dashboard. So we have developed a lot of dashboard, but we can also customize this dashboard if you want, if you have special questions. I can show you the dashboard about for instance, what we have detected on the video. So again, sorry, it's in French, but we can translate quite easily. So we have developed many dashboards. You can see where the detection have happened. You can have the numbers of species, 38. You can have the name of the species. It's in French, but I can translate. It's not the African savanna. It's a vineyard, so that means the thing you detect is the boars, deer, mice, cows, and so on. You can have the repartition. You can have the detection side by side. You can have the detection around the year. You can have a vision of all the detection in space and so on. So we have developed a lot of dashboard for the cameras, for the sound. Sound is quite interesting. But today we always use software for the recognition of the signal, and we use software based on artificial intelligence, and we use software developed by universities. For the birds, we are using the software developed by the Cornell University, which is the most impressive. So at La Perruche, we have something like 136 species. It's huge. I'm going to compare you to other seats. It's huge. It's very impressive what the owner shall obtain with all he has done for the birds at La Perruche. So here you have again the word clan and so on. So this is the platform, and after that, we can use the platform to make analysis. And today we don't integrate the analysis to the platform because they are customized analysis. So I'm going to show you some slides about the first analysis we made in last October at the half of the collecting season. Now we are going to make again the analysis just to show you the kind of thing we can do with this data. So first, we need questions, okay? We need questions to propose an experimental plan and then to execute the plan. So at La Perruche, we've got 5 questions. The first question is what's about the soil? Shall the owner make a huge effort for the soil with green cover, with green manure, and he have no idea what happened in the soil, and he asks us, what is the biodiversity, the quality of the microbiota of our soil. The second question was about the fauna. He has done a lot for the fauna in terms of habitat, for the mammifer, for the bats, and so on, and ask us what is the population of the fauna. With two focus. The first one is on the birds, because birds are considered like auxiliary for viticulture, and bats. And the last question was about biodiversity and indexes of biodiversity. Could you produce some indices in order to monitor what I'm doing and perhaps to compare my vineyard to other ecosystem? Okay. So for the first quick questions, we use two different technology. The first is a technology developed by a French company which is called Terramea. And this technology is based on the technology which is called flux cytometry. It's the technology that you use when you make a blood test in medical device. It's a technology that counts your red blood cell and your white blood cell. Okay. In this case, we take a soil sample and we count with this technology the bacteria, the fungi, and the protists. Okay. It's the three column you see there. And the company Terramea make a special index, which is a good equilibrium between the three main phylum. And if this index is high, that means that you have a good equilibrium for your vineyard. Okay. And we make 8 points at La Perruche. It's huge. You don't need to make 8 points. It's because we want to test the technology. Okay. To summarize what we obtain, the soil of La Perruche are in perfect state for the microbiota. It's the best you can have. There is just one point here, which is called Garenne, where the microbiota is less in good health. And the owner is not very satisfied there by the terroir and by what is obtained, and is still changing the way is practicing this green cover. Okay. The other technique we have tested is more standard, is the eDNA technology, and we test that with a French laboratory based in the south of France, from the CNRS. Perhaps you know the CNRS, which the very familiar institution in France. And we have made some, so it's very simple to understand. We take a soil sampling, we sequence the DNA inside the soil, and then we obtain a table with all the species and the quantity that we obtain. Some results that we obtain, which are now very known by everyone. So here, I'm sorry, you can see the 8 points in the vineyard, and here it's a percentage of abundance. And here you have 3 colors for 3 phylum: Archaea, Bacteria, Eukaryote. We are Eukaryote. Huh? So as you can see, it's blue. Huh? It's almost only blue. That means that when you use eDNA, you see only bacteria. It's very difficult to see other things. Or you have to make a special amplification, what we call barcoding. So if you use this technology, you have to work on bacteria. The other results, which is quite known because there is a big, there is a very important publication in Nature about that, is the fact that if you just look the species, the number of species per samples, what we call the specific richness, it's all the same everywhere in the field at La Perruche, even in the forest, is exactly the same. You have between 6,000 to 8,000 species. And the paper in Nature have demonstrated this thing is true for every kind of soil in agriculture, conventional to organic, to every site. So the taxonomic diversity is not differentiating factor for the soil. You can find other way to differentiate soil, and we are working on that with CNRS. I give you just one example. Here we have just the graph for the 20 most present bacteria in the soil. And as you can see, there is a difference here. Here is the forest. So you can differentiate the forest from the other things. And here is very blue because in forest, you find more bacteria which are fixing nitrogen than elsewhere. So that means that if you want to work with the DNA on soil, you must look for what we call the functional diversity. Okay. So the conclusion for us, because the question was, how was my soil? So with the flow cytometry, we demonstrate that we have an excellent biodiversity typical of agricultural soil with mixed use land, like organic viticulture. There is a point where there is a tiny difference with the other point where we have a readjustment of plant cover. And for the other technology DNA, the initial analysis have not been able to fully answer the question. And we are working with the CNRS because since we have made 4 oversampling during the 4 season to go through the problem between taxonomic differentiation but we have answered to the question, to the first question. The second question was about the wildlife in video. So we have 38 species. I won't comment. I don't comment that. I'm going to show you through the indexes what it means. But I would like to speak more about the birds with the sound detection, because in this case we have enough data to start what I explain you, what we call the biostatistics. So here you have this kind of graph where we have tested infrastructure. You can see forest, edges, or environmental the parameters such as the rain or the temperature. And we are a biostatistician to see if they have a positive effect, it's blue and it's up, or a negative effect, it's red and down. So for the bird, all the bird detect with the sound at La Perruche, the statistics show that the forest have a negative impact and the edges have a positive impact. I think you know that, but we are very proud to show that by statistics. Because we have many data with the birds, we can go down to species. We have enough data to make this same approach species by species for 50 species of the 136 species we have detected. And I've here choose the example, which for us is very interesting. Here we have what we call in France Alouette lulu. I don't know the name in English. It's a bird which is in way of extinction. It's protected in France. And if you want to show to your customer that with your practices, you make conservation of a bird, this one, at La Perruche, we have shown that this bird loves the forest. When it's rain, it don't like the edges, and it likes when it's hot. Now, if you want to focus on the fact that birds can be auxiliary for culture, here we have I forget the name, mésange. You remember what's the name, Patricio, in English of mésange?
No, I don't know.
Yeah.
In a few minutes.
I think you recognize on the picture. So it's a good auxiliary of culture because they eat worms, grape worms. And in this case, they don't like the forest and they like the edges. So it's a way to see what kind of infrastructure you want to put at La Perruche. This statistical are good for La Perruche, okay? And we have done that for 50 different species. So we have answers to the question for the birds. Many species, very good job. It's very great for the birds. The fourth question was about the bats. How many species? And there was a special question for the bats because bats need to have points to trees and so on to move because they use the echolocalization, and sometimes the bats are not going on the vineyard. So here the question was how many species I've got, and do these species are going inside the vineyard. So it's why we make here measurements, is the number of detection we've done around the vineyard and inside the vineyard, and the answer was very good. Because here you have the central point of vineyard, and you can see we have many detections. That means the bats are moving to the center. And why? Because Charles, the owner, has planted edge of fruit trees through the vineyard to give them point de repère, forget the name in English, to move through the vineyard. We have 16 species. To give you an example, in Aquitaine, near Bordeaux, we have 22. The maximum is 22, 40 species with very special, but it's 22. So the biodiversity is not huge, but it's very interesting. And again, because we have many data, we can make a biostatistical approach, species by species. So here we have two different bats, the great murine and the murine of Daubenton. And we have very evident results, but demonstrate by statistics. So as you can see, the forest is positive, the building is positive, and the edges are negative for the bats. So if you want to have bats on your vineyard, and if you want the bats moving on your vineyard, please don't touch the trees and keep your building for the bats. Okay?
Look,
Last point.
2 or 3 minutes.
Okay, so now I'm going to show you the indexes. So for the indexes, it's very simple to understand. We have the data, so we can calculate the indexes you want. Okay, we don't have a philosophy about that. Here we have to calculate Shannon index, species richness, and Simpson index. We have discussed that we are specialists. They are very standard index, but we can calculate over index because we have the data for that. To show you what we can do, just this short animation on the Shannon index. I think it's going to be interesting to that. So here we calculate the Shannon index for Château La Perruche with the sound recording and with the cameras. So we have two numbers. Don't mind mean nothing for you, but we can compare to other sites where we have similar. So here, Étréchy and Château-la-Vallière. So Étréchy is a site where we are measuring. It's near a small city, near a forest. So Château-la-Perruche is better than a natural zone near a city. And Château-la-Vallière, you can see, it's an hostel near a natural zone. So as you can see, Château-la-Perruche is better than Étréchy and Château-la-Vallière. And because we have the open data, we have calculated the Shannon index at Langoiran, that means around La Perruche. So as you can see, La Perruche, it's better than La Vallière and Étréchy, and it's just under the global ecosystem. That means that it's not so far from the best you can do, because the best you can do is this number, because it corresponds to all the ecosystem. Okay. And perhaps I can stop there to see you the potential of this kind of approach for vineyard.
Okay, thank you very much. I don't know if some of the members of the committee, if they have any question. I don't see for me, it's a lot of information, but I don't see.
So it is a lot of information, yes. So I've got a question. So in order for you to rise a profile of biodiversity of a site, you need lots of databases, as I guess. I mean, from your sensors, birds stinging or recognizing vertebrates. So, which I understand in France, you've got lots of those free databases. What about going to some new places? I'm not sure in Chile we have something like it.
So again, we use three kinds of sources: open data, sensors, and your data. Okay. Sometimes you have some data. even some biodiversity analysis done by a naturalist. Okay, so the first thing, if we work together, could check that. Concerning the open data, sorry for that, because I could ask to my data manager to do that, but we can check how many data you get in Chile, because the JBIF open database we use is a worldwide database. Okay, and for the environmental information concerning the soil nature and so on, there is a database from Microsoft with 200 open data, and I think we can check if it's covered also Chile, because it's a worldwide database. It's concerning the open data. After that, you can also have sensors, which are very similar and not so expensive, and you have your data. Your data are very important. So we are not so anxious about the fact to move from France or Europe about that.
Thank you.
Okay, any other question? I don't see all this. No?
It is very interesting, by the way.
Thank you.
Thank you very much. Look, is it possible to share the presentation also? Because
Yes, of course. Yeah, I can make a PDF. Yeah.
And I think it's a very good opportunity as a first conversation with you and with our members of our committee. I want to share the information, the video, and then we can continue if there are some interest in order to implement or to see in the future any projects or pilot projects in order to measure the biodiversity, because it's crucial.
Okay. And to give you an information, we are starting to work with people outside France. So the fact that the project is far away from base is not a problem. We are going to start to work with Moldavia.
Great.
Okay.
So thank you very much once again. We're gonna be in contact here.
Thank you very much, everyone.
Yes.
And thanks, Patricio, for that. Bye-bye.
Bye-bye.
Bye.